So far in this class, we've been really looking at how life is built up from the small to the large. And we're getting close to rounding out the whole picture of how each part is built. We've looked at how amino acids form protein chains, which do most of the work inside the cell. We've looked at how the four letters of DNA and RNA help make those longer molecules. And we've also now looked at membranes and how membranes are built from lipids, and that membranes form enclosures which make up all the different organelles inside of the cell. So what we're going to look at now is how polysaccharides are built from smaller sugars and what polysaccharides are even for. Some people might think of these as just little doodads and decorations that are on the outsides of cells and in different proteins that kind of decorate the outside of glycoproteins. But they're very interesting molecules, and they have a lot of special properties. The biggest property about polysaccharides, in my mind, is that they form branches. So they don't just form a linear chain the way that protein and DNA and RNA do, one letter right next to the other. They can branch out. So one letter of the polysaccharide alphabet can connect to two other letters, and then those can start forming their own little chains, and those can branch out from there. Well, this obviously makes for many more combinations. If you have 10 letters in your alphabet, and if you have a length of 1, okay, then you can have 10 different combos. Okay, you can put your 10 letters in that one little spot. If you have a length of 2 letters long, then you can put all your letters here and all your letters there, and actually that's 100 combos, as you can probably imagine, from 00 up to 99. Okay, and if you have 3 letters, it's 1,000, 4 letters, etc., etc. That's for linear things. So for DNA that has 4 letters, if you have 6 spaces, you can get over 4,000 combinations. And that seems like a high number. But if you had 4 sugar letters and 6 spaces with 5 different ways that you could branch out, and that's pretty typical for a sugar, you can even have more than that, then instead of 4,000 combinations, you can get over 1,000,000 combinations. So in the same space, with the same number of letters, you can get so many more combinations. It makes it possible for each cell, and there are more than 10 trillion cells, for example, in a human body, to be different from one another. It's also possible for each human, and we know there's about 7 billion of us here on this planet, to be different from one another. Even identical twins can be different. There's no problem with that. Now, what does this mean? It means that finding a perfect match for organ donation seems impossible, right? If everybody has different sugars on the outside of their cells, it seems absolutely impossible. But thankfully, we know that you can get an organ donor, and perfect matches are not exactly required. So that's good news. But this getting things on the outside to know what parts of the cell are self and what parts are enemy invaders is very important, and that's kind of what the sugar does on the outside of cells. An activity in your mind that you can think of, a mental activity, is what makes a peanut M&M? What kind of calorie types are listed on the label of your peanut M&Ms? Well, if you think about it, there's fat, there's protein, and there's carbohydrates. Okay, well, that's how it is for almost all foods. In peanut M&Ms, how are those 3 types of calories organized? Where's the protein? Where's the fat? Where are the carbohydrates? Well, if you think about it, the protein is mainly in the peanut, which is down there in the center. The fat is in that little chocolate layer that make the peanuts M&Ms so tasty. And then the carbohydrates are everywhere, but especially in the outer candy shell that keeps it from melting in your hands. So that's exactly the arrangement of a cell. The protein is kind of in the middle. The lipid has that fatty layer that keeps ions from flowing in and out. And on the very outside, there's a decoration of lots of polysaccharides. What is the advantage for cells to put them in that order? Well, it's not too different from the advantage of getting peanut M&Ms. Keeping the candy shell on the outside prevents it from melting, and on the cell, keeping the candy outside shell, it makes sure that the cell is very happy to be in an aqueous, watery environment so it doesn't stick to too many other things. Now, sugars are all over the place, and one place we see them is in blood typing. Blood drives that we all are aware of keep us conscious of the fact that not everybody's blood types, even for the red blood cells, are the same. There's people who have A positive, people who have O positive, AB, all that stuff. And do you know what your blood type is, and do you know what that means? Well, if you go on the figures and look at what those pictures look like, you'll see that an O-type blood, an A antigen blood, which is on people who have A antigen or AB blood, or B antigens, likewise, for those people who have B in their blood type name, are very similar. The ends of those polysaccharides are the only things that are different. A antigen has something called galactosamine on the very end, whereas B antigen has glucosamine on the end. And O has neither one of those. So they're very similar, and yet without that one little thing, giving a person the wrong kind of blood could lead to a terrible reaction and end in death. So these little tiny sugars on the outside that tell self from non-self are extremely important, and it's a good thing we know how to blood type people so we know what kind of blood to give them. If you're looking at the textbook, this is a good time to review the leaf, What are Carbohydrates? It's a general overview of how polysaccharides are structured, and you can do a little bit more reading on your own if you're interested in blood typing and organ donation.